respected faculty member, my dear students, and all other viewers and seekers. A very good morning and warm welcome to all of you. As a part of IQAC activities of Surendranath College, the Department of Microbiology has organized several webinars and virtual training sessions earlier. To continue with our effort of such outreach activities, we have a series of webinars and podcasts lined up for you. Today, we are happy to air our first international webinar by one of our proud alumni, Dr. TARUN KESHWANI from Harvard Medical School. But before going to the lecture, I would like to first request our Principal Sir, Professor Indranil Kaur, to share his thoughts and valuable feedback.
Very good morning to you all. It is really a wonderful occasion when we hear our own alumni speaking from, and if we talk in that way, it is Sat Samudra Dure. We are talking from we are listening from a person from far off area, and we are really proud to be part of this institution where we have large number of wonderful and illustrious alumni around us. To talk about this webinar, actually, I should tell that I was associated with the ICMR and I was dealing with infectious disease. I was dealing with vector-borne diseases particularly. So it is really interesting for me to hear all this. If we talk about the infectious diseases, I'll just narrate a few lines. Then I think Tarun will be taking up.
Sure.
Infectious diseases defined as the disorder caused by the pathogenic organism. These are bacteria, virus, parasites, and fungi. And they can be directly infected or can be indirectly through the vectors. These typically, actually, it was initially when we were getting infected with the various types of diseases, the invention of penicillin brought us a relief. We started thinking that it almost appeared that the infectious diseases will be almost over. But this problem again cropped up when we got the genetic selection of the microorganisms where we find that the resistant varieties to get selected. And ultimately, again, we had to give it a second thought. And finally, we are really again puzzled that how to tackle these organisms and save the mankind in different times. I think Tarun will be a wonderful person working in the best situations. So let us hear from him instead of me telling you all these things. Oh, I think Tarun, over to Tarun now.
Thank you, sir. Thank you for the nice words. First of all, I would love to thank my department people where I
may I interrupt, Tarun? May I interrupt, Tarun?
Sure.
Shall I introduce you first?
Yeah, sure.
Yeah. So thank you, sir, for your inspiring note as always. Now, I know all of you are eagerly waiting for today's lecture by Dr. KESHWANI. It is a great pleasure and honor for our department to have him with us today. I take the privilege of introducing him today. Tarun did his graduation in microbiology honors from Surendranath College in the year 2006 and followed by his MSc and MPhil from Kalyani University. He had earned his PhD in zoology from University of Calcutta in 2016. During his PhD, he worked extensively on the role of dendritic cells and T cells interaction during malarial infection. Tarun has received several international and national fellowships and awards, including the prestigious Raman Charpak Fellowship, an Indo-French 6-month predoctoral fellowship, Elsevier Award for Contribution in Parasitology by Australian Society of Parasitology, Gold Medal from Indian Society of Parasitology at Benares Hindu University. He has continued his research as postdoctoral scholar in Institut Pasteur at Lille, France, where he was a recipient of Marie Curie co-founded European Union 2020. After a short stint in France, he moved to Department of Medicine, Albert Einstein College of Medicine in United States, where he was involved in investigating cerebral malaria, a severe complication of plasmodium infection. Currently, he is appointed as a research scientist at the Department of Medicine, Massachusetts General Hospital, a part of Harvard Medical School in Boston. So without any further delay, let us welcome our speaker of the day, Dr. TARUN KESHWANI.
Thank you for such nice words. I mean, it's really honor to be part of this college or the place where I learned a lot of things. I made a lot of friends there also, and we saw a lot of faculties also. I mean, the current faculties, and also when we joined, there were a lot of transitions. So it's really nice way to give something to the department and learn more from my PIs. So can I share my slides now?
Yes, sir.
Yeah, I'm looking for the link, which is minimized now. Yeah, you can see my slides.
Yeah, they are starting.
Okay. Shall I start?
No, they are coming, Tarun, actually.
Okay. It's showing that screen sharing is paused.
Oh my God.
Okay. So what I will do, I will stop and start again.
Yeah.
I think it's good now, right?
Yeah,
Yes, yes.
Okay. So
Go to slide. Huh.
perfect.
so before starting, again, thanks to Principal Sir and all my mentors and all the students who are spending their 1 hour. If you are feeling bored, or if you want me to slow it down or explain it in a different way, please put some message in the chat and I will try to explain in a different way. So first of all, I try to think what title should I present or what topic, because these days people are working and presenting extensively on COVID stuff. Then I thought that as I'm working on malaria and I had some experiences working on Ebola vaccine stuff, so then this title came up, a review on emerging and re-emerging infectious diseases. So currently I'm working here at Harvard Medical School and Mass General Hospital. My topic is a bit different here, but I'm working, but I will explain that later on. So I saw that similar kind of slide was there few minutes back. So thanks for that. So first of all, I would love to present a global example of emerging and re-emerging infectious diseases. As we know that before COVID, there were some outbreaks of SARS and MERS and other stuff. For several decades, there have been some outbreaks here and there worldwide, but those were not taken seriously, I will say, for the people from different nations where it was not happening. Why I'm saying that? Because people did travel to places before knowing that what are the local outbreaks in those places, right? Like in Southeast Asian countries, like many before COVID, when the SARS and the MERS came, it did try to spread, and luckily, though there were a lot of setbacks, but there was not huge global expansion of those. They were clustered in some places, and we can see here several examples. So in Lancet 2008, this topic raised up, right? Like Ebola, influenza, yellow fever, malaria, plague in India. A lot of stuffs came up. So this is just an example worldwide. And what important stuff came up in last 2-3 years, though it was there, that the connections, international collaborations, and how that will be helpful in near future to resolve any further pandemics or other emerging and re-emerging diseases, right? So this is a graph or cluster from Washington University. So they have a lot of networks in US and worldwide. what they do is that they share data, they share information, and try to upgrade the civil society with what is going on with the infectious diseases and other stuffs. So before moving on to the main part of the slides, I broke down the slides into 4 parts. That is the fundamentals of emerging and re-emerging diseases, and then the immune response triggered by vaccination in Ebola, emerging diseases like malaria and co-infections, which Principal Sir was mentioning, that things got changed with the infectious diseases and things are still changing with the co-infection stuffs. And then the basic and translational human immunology and how future things are molding up in a way. So first part is the fundamentals of emerging and re-emerging diseases. And so what is emerging infectious diseases? According to NIAID, National Institute of Allergy and Infectious Diseases, emerging diseases are commonly defined as the outbreaks of previously unknown diseases, sometimes known diseases that are rapidly increasing in the incidence of geographical range in last 2 decades, and persistent of infectious diseases that cannot be controlled. For example, as I mentioned before, the HIV, Zika, West Nile, dengue, SARS. And what are re-emerging diseases? So re-emerging diseases are those due to reappearance and increase of infections, which are known, but had formerly fallen to levels so low that they were no longer considered a public health problem, like malaria. I mean, in some places, India and other parts, we think that malaria is gone, but still malaria comes back, here and there. Like TB, it still persists. I mean, when we do, medical tests, when we come to US, many people get TB positive because so one doctor told me once when I was doing this test that if you are not TB positive staying in Kolkata and working, then you are not working actually. So people do get this, right? But those are not virulent strains. So actually in 1995, WHO did made this establish this department called Division of Emerging and Other Communicable Diseases, Surveillance and Control. But I doubt in India we have thought of this. Maybe there are some new departments working on this. Like, when people travel in US or in Europe, they do take travel medicines and they do take vaccinations. So this thing changes a lot of things actually. Like the primary thing is that they stop in spreading other newly infectious diseases. So traveling abroad can put risk for infectious diseases, right? That not widespread in the United States. Learning how to reduce their risk of getting these diseases are also important. Hello. So yeah, so sorry, the slides are a bit stuck. So the important part is that we love to travel, right? But we hardly think about this aspect. I mean, I'm speaking about before COVID, not even during COVID. I mean, we think that what food is available there, where we can visit. We think that where we will spend more time, where we will spend less time, maybe in, wild areas, jungles. But we hardly think that what are the local infections there, like what we are aware of the unaware of the potential hazards in different parts of the world. And many of these newly discovered infections have actually been in the existence for a long time, but when people started traveling more for several centuries, things spreaded more, right? Especially the infectious diseases. So how can we reduce the risk from infectious diseases to spread? We have to see a travel medicine specialist or healthcare provider familiar with the area where we are moving to, and get the immunizations and take preventive medicines recommended by the healthcare provider. So, I mean, this thing is also new for me, but I try to formulate the thing in a way that how it will feed into our current area of research and health. So this is a triangle disease concept adopted from McNew in Plant Pathology, published in 1966. So this is a whole modeling which reflects the constant struggle of microorganisms to survive. 3 important factors are there: the host, the environment, and the pathogen, which leads to the disease and brings the outcome. There are other factors which leads to this is the deforestation, a deep rainforest, modeling changes in ecology and biodiversity, and which leads to the shifts in ecosystem and pathogen exploits new ecological niches, which leads to the new viral strains. Multiple species, Leishmania, Plasmodium, and Toxoplasma persist in this kind of environments. Like in Amazonian river basin, you will see this kind of niche, which leads to multiple differential immune responses, chronic asymptomatic carrier, and also reservoir for viral transmissions. So infection outcome is dependent on complex and multiple interactions for sure. I mean, it's tough for a researcher to understand each of the part uh, in detail. So that's why this collaborations, networking, all these aspects are very important and helpful for the society. Next, I would love to show, the slides are moving a bit slowly. Yeah, so before moving on to the other parts, sorry for the technical glitch. This is a slide explaining the major emerging and re-emerging infectious disease outbreaks from 2002 to 2020. We can see that starting from 2000, gradually there are a lot of different emerging diseases, but you can also see with the colored bars that the length of the diseases means the time period this stayed, before omitting from the system start increasing. Like if you see for this Zika virus, measles, Ebola, again there was a second outbreak in Democratic Republic of Congo after this, and measles also in MERS. It's still sustaining, and then in 2028 COVID, beginning the COVID started. I don't have the updated slide for 21. As all of us know that it's still persisting. I hope things will be better in near future. So I mean, so the point is that the emerging diseases still persist here and there, but how to deal with those stuffs? And I will come to those parts soon. So that's why I kept one part where how the immune response are triggered by vaccination, in Ebola. And so before moving on to that, there was a Ebola outbreak this year also. I think we hardly noticed that part, but on 7th of February 2021 in North Kivu province in Democratic Republic of Congo, there was Ebola virus disease outbreak. And in 2018 also there was an outbreak. So after genetic sequencing analysis, scientists predicted that this outbreak is linked to the 2-year-long outbreak, and luckily the outbreak was declared over on May 3rd, 2021, after several efforts from the research scientists and doctors working there. So how does Ebola virus work? It's mainly originated in the African region, and it was first discovered in 1976 near the in the Democratic Republic of Congo. Since then, the virus has been infecting humans from time to time and leading to outbreaks in several African countries. And it's tough. I will explain that later, that it's tough to reach in those regions because some vaccines need proper storage conditions, and it's tough to maintain the vaccines there, and they lose their efficacy. So it's very tricky part. Anyways, so the source of the virus is from the fruit bat, and it transmitted to humans from the gorilla, chimpanzee, and the duiker. And when human consume the meat, or or from the indirect contact, like the secretions, organs, or the other part, consumption of the infected bushmeat, touching objects, and it caused huge damage. I mean, incubation period is from 2 to 21 days, and death from disease often caused by multiple organ failure. And now we are well aware of this multiple organ failure part. In COVID also we have seen that. And it targets mainly the liver, the endothelial cells from the blood lining, and the phagocytes blood cells that absorb foreign particles. So this is a brief. I'm not going into more detail about each pathogens which I'm describing. I'm trying to make it more subtle and simple so that we get the message and we discuss further about the outcomes. So here as I mentioned, I will discuss about the Ebola immunogenicity and the pan-Ebola vaccines. Why pan-Ebola vaccines? Because where I worked in France after Institut Pasteur, I stayed there for 1.5 years almost. And there I was lucky to get this opportunity to work on the Ebola vaccines. There was a huge conglomeration in Europe. So before moving on to that, this is a cycle of the symptoms of Ebola virus. It's typically through a human being. The first symptoms comes in about 7 to 9 days, and it's sustained till 12 days with the loss of consciousness, seizures, massive internal bleeding, death. So what is the percentage of survival in this? I mean, it's very critical here because in 7 days, if a person survives till 7 days, there is a 30% chance of surviving. And if a person survives till 3 weeks, there is at least a 90% chance. But as I mentioned, that there's a multiple organ failure and it's very tough, and for most of the patients are infected persons to come out from that infection, and it's very contagious also. So what we did there in France, Nice, there were two formulations. Why I'm showing this slide? Because the formulations are very important for the vaccine development and the sustenance of the antigen at the place where we are injecting the vaccine is very important. So this formulation, like the glycoprotein, Ebola glycoprotein, and the nucleoprotein, I'm not able to explain more detail about this because these are company stuffs, but I'm just explaining what this glycoprotein and the nucleoprotein and glycoprotein combinations, which are called long synthetic peptides, which has multiple features like it expresses the CD4, CD8, and B cell epitopes, whereas the glycoprotein work mainly on the long-term B cell responses in increasing the B cell responses, and the GP and NP, which is a combination and named as LSP further, they have a strong and long-term CD4, CD8 T cell responses. They helps in inducing that. So how this system works, right? Immune response triggered by vaccination. So there is a priming step, as we know, and then the boosting step. So three factors are important here: the formulation, the delivery system or the adjuvant, the antigen dose, and the route. So that's why it takes time for the researchers to figure it out, that what is the best route and formulation and the dose to understand the immune mechanism, and then further move into the different phases of trials before working on the various model systems. So the immunostimulatory properties of most adjuvant relies on three mechanisms of actions: prolonged retention of the immunogen at the site of injection. So that's why most people might remember that when we were getting the vaccines, they were taking some times to prepare the formulation and they were injecting. So it should be prepared at that time. And then activation of the innate immune system through recruitment, activation, and maturation of dendritic cells, as well as other innate leukocytes is very important. And then polarization of the antigen-specific immune B and T cell responses. So how this works? So this is a summary of all the stuff at one place, just trying to make things short and have more information. So when the antigen dose or formulation has been administered, so the local cytokines, and the antigen competition, the vaccine when administered, so the DC interacts with the T cells, it leads to the differentiation of Th1, Th2, Treg 17, depending on the cytokine microenvironment. And also this leads to the development of short-lived plasma cells and the germinal center B cells through Th follicular, leading to the long-lived plasma cells. So they have the respective importance in the immune system, the short-lived and long-lived plasma cells, depending on the vaccine strategy and the vaccine formulations. The expression of the development of the long-lived plasma or short-lived plasma leads to the better efficacy of the vaccine, which I will come later on. And then it leads, goes to the blood and the periphery, where there is antibody production, the plasma cells comes in the blood, even the T cells. It then moves to the development of the memory response when we boost, when there is another dose, which again, there is a differentiation of the T cells and central memory in the lymph nodes, and B and T memory cell productions, and in tissue there are effector memory cells. So all this stuff goes together, and that's why I'm saying it's tough to understand the whole immune mechanism, which day to day they are more complex, the immune responses. So I will try to explain the experimental strategy, what we obtained there, to understand the immunogenicity of Ebola vaccines and selection of optimal prime boost strategy. So we had two formulations, as I was mentioning before. So we also use emulsion for the first vaccination on 0 day in the wild type C57 mouse. And we also use the adjuvants here as a Hiltonol, CPG, or MPLA, which are different adjuvants like TLR3, TLR4, 8. And the emulsion is also important because it should be the slowly released effect of the antigen. And then on 14th day, there was again second vaccination of the same combination, respectively, the recombinant GP and adjuvant and emulsion, and here LSP adjuvant and emulsion. And then after 28 day, which is the effector phase, we took the serum, the cytokines, and the B and T cells, measured the cytokine staining cells through intracellular staining and IF and gamma we did. So we collected all these respective organs and stored the serum also. After that, we waited till the memory phase. So we had different batches of mice. So few we sacrificed here and we kept other mice and we checked the memory phase. So that's why when we plan these experiments, initially we have to plan for all this 75 days and we have to have all the resources. And also we have to book these days on time. So that because these days are very long working days. So we have again similar kind of sample collection on those days, and what we obtained from this formulation, as I mentioned, the strong B memory response, and we obtained strong T cell memory response from this. So what does this suggest? That the strong B and T memory cell responses, if we use this combination of GP and LSP based vaccines for Ebola. So in summary, what we observed, I didn't show all the data here. These are just schema. And we saw that the TLR9 adjuvants and showed better or similar response with TLR3 than TLR4. We saw that there was increased CD4 T cell responses, expansion of CD4 T cells, Th follicular cells, and decreased Treg cells we observed. And we also observed that there were long-lived and short-lived plasma cells in our system during the priming event, and we observed that there were high antibody. And in memory cells, we observed the increased T central memory markers, which I am not explaining here what are the markers. And B and T memory cells were high in the memory phase also. So, but the interesting part here is that what makes a vaccine more efficient? So we have to think, which is tough to think before starting any experiments or designing, because after we get the data, then we discuss with because mostly in this kind of studies, the companies and the lot of institutes are working together. So timing is very important, and arranging all the people at same place is also tough. It's always complicated with this kind of studies. But at the end of the day, we have to see whether our vaccine was targeted to memory B cell subsets versus non-targeted vaccination. So we know that lot of vaccines don't go through the pipeline. They stop showing any efficacy because of these factors. Like if we don't get a long-lived memory population in infection-specific localization or effective antibody response compared to the unmaintained memory response or improper or ineffective activities, then it's very tough to get a good vaccine outcome. So role of particular memory B cell subset in a vaccine, the localization, type, and magnitude of the antibody response, which we are all aware of now, well aware of, and the maintenance of the memory B cell subset populations are very important for predictive efficacy. And also, as I'm mentioning, the memory responses. So there are several subsets of memory T cells, and central memory T cells are the prime memory T cells among those, which I showed prior in the schema. They produce IL-2. So this slide shows very succinctly that the effector memory cells express lower IL-2 but higher IF and gamma and cytotoxicity. So we get this information in the prime phase, but in boost step or the memory step, we see that the central memory cells differentiate and they express higher IL-2 and almost less IF and gamma than the effector memory phase, and they are less cytotoxic. So this kind of study gives us the information that whether a vaccine will be more effective or not. So after this step, I will move to the emerging diseases chapter, where I will discuss about the malaria and the co-infections. So, in light of the current outbreaks, this is really a worry I mean, this was published last year on some article that malaria could have a resurgence due to COVID-19. I mean, it's the simple reason is that that only mainly children will be impacted, but most of the health services were occupied with the outbreaks, which was need of the hour, but it was tough to send the help to the respective places in the tropical countries where there is and which leads to the disruptions of the burden on malaria. So this is with not only with malaria, many other diseases also, those were a bit neglected, but that was not need of the hour maybe that time, but maybe in coming few months or years there might be outbreak of other diseases also, which is very bad condition. So there was a case last year in late 2020, and healthy woman returning from Tennessee after a mission trip to sub-Sahara African country. She was she got fever, chill, sweats, and headaches, and she went to the emergency room and was tested for COVID-19, and coincidentally malaria both, because she traveled to those places and it was, I mean, she got that malaria. But it's very complicated that time because doctor will think that she has COVID-19, but her condition rapidly worsened, and sadly it was too late. So this devastating case of the woman from Tennessee is a reminder that our work is not done yet. I mean, there is a lot of things to, I mean, there are a lot to fill, right? There are a lot of blanks still left. So coming into cerebral malaria, that there are almost, I mean, 228 million cases worldwide, and that's almost, I mean, every second or every 2 minutes, one child dies from malaria in Africa. And 70% of the malaria deaths are children under 5 years old, and they mainly die with the cerebral malaria, respiratory distress, metabolic acidosis, and anemia. So what we know about the immunology of malaria, there is always a tenuous balance between the parasite host and the immune system. I mean, whether there is a protective immunity or trigger harmful responses, it's still unclear, because there is always a group of scientists discussing the balance between pro-inflammation or the anti-inflammation, which one is beneficial, which one is detrimental. And also it's difficult to unravel the mechanism of protection of pathology in humans. So that's why we need model system also. So this is a basic life cycle of the malaria infection. And I would love to go through it quickly. So we work mainly on the erythrocytic stage, because this is a stage where we understand that there is a malaria infection, because the chills, fevers, and the other symptoms are understandable at this phase, the blood stage, when the parasite comes into the blood stage. But normally, when a parasite infects a human, the sporozoite goes to the blood, and from there it goes to the liver. After mitotic replication, the liver cells rupture and the merozoites released, and then the parasites are again infected into the blood, and it goes through the cycle of, the seizoan, trophozoite, and the wing phase, and then wing phase again when they were, and we have the gametocytes also. So when the mosquito again bites to the infected human, then the parasite again goes through the cycle, and again the infected mosquito can bite to an uninfected human, and in this way the cycle goes through. So that's why, as it's a multiple phases, it's very tough to make vaccine for this kind of infection. So still people are working on this, but it's very complicated. I mean, there are several groups working on the liver stage, why the parasite goes into the liver. Many people works on the erythrocytic stage, and how the parasite from erythrocytic stage goes to the brain and causes a blood-brain barrier disruption, which leads to the cerebral malaria. Many people works on the mosquito stage, the gametocytes phase, and the midgut from oocyst to the development of sporozoites. So a lot of stuffs are going on, I mean, worldwide. So we work mainly on the intraerythrocytic, the sexual symptomatic cycle. So it's a vector-borne disease, as we know, but it's caused by the protozoan parasites of the genus Plasmodium. And according to the disease severity and rare and mild cases, this is arranged. The falciparum is the most severe, and then also vivax, oval, and malaria. Malaria is mainly in the non-human primates. So human don't get that infections. And according to WHO definition, the cerebral malaria, which leads to coma and peripheral parasitemia. So this is a mosquito with the, you can see the infected blood mostly there, ready to bite. So coming back to the brain, because as we are discussing about the cerebral malaria, this is a brain parenchyma where we can see during Plasmodium falciparum infection, which is acute phase, we can see the brain structure here. The neurons is adhered here. This is the lumen of the blood vessel. Tight junction proteins are expressed here, endothelial cell, astrocyte. And if we go further detail into this part, the tight junction part, we can see that there is a nice blood barrier which is intact in a healthy human being. The endothelial cell, the basement membrane, and the tight junction markers. And we can see that there are lymphocytes, blood lymphocytes, monocytes, neutrophil circulating in the periphery, and the astrocytes and microglia inside the brain. So what happens during the Plasmodium falciparum infections? That there is a disruption in blood-brain barrier, which I will show another schema. There is acute inflammation. The CD8 T cells infiltrate into the brain, activation of the glial cells, astrocytes, and a lot of chemokines and pro-inflammatory cytokines play an important role in the acute infection causing cerebral malaria and coma. So it's very hard to pinpoint which one is mainly important for the coma, but that was my one of the project last year at Albert Einstein, where I was working on the metabolites role in cerebral malaria, neurometabolite, I will say. So this is a mechanism of cerebral malaria. So what leads to the blood-brain barrier breakdown is that when there is an infection, the parasite goes to the endothelial cells, and there is a clumping, autoagglutination, rosetting of the parasite, cytoadherence to the endothelial cell walls, which leads to the obstruction of blood flow, and which releases a lot of microparticles and toxins and leads to the hypoxia and altered blood flow, means the reduced oxygen level. And that leads to the blood-brain barrier disruption and recruitment of leukocytes and platelets, which then adheres there, and release of cytokine occurs. And this leads to the multiple seizures and neuronal damage, and finally hampering the tight junction proteins and leading to cerebral malaria and blood-brain barrier breakdown. So the key questions here, I mean, what is the pathophysiology of severe disease? How do people living in malaria endemic regions become protected from severe disease? That's a big question, how they become protected. And this cannot be answered fully by observations of infected humans, because it's tough to understand what is going on in the brain physiopathology. But now these days we have MRIs where we get the information about the ultrastructure of the brain, but it's tough to understand the mechanism by observing the infected humans. So that's why we need mouse models. So there are well-known experimental cerebral malaria models. So this one is the traditional one, the C57 black 6 mice. And also the good thing is that we can also work on the erythrocytic stage and the sexual stage both, because we can infect these mice with the sporozoites directly obtained from the infected mosquitoes. So mosquito culture can also be done in the specially ethically cleared mosquito culture rooms. So all the institutes, it's tough for the institutes to get the clearance, but we can have the the oocysts and the sporozoites from the salivary gland, and we can keep the mouse like this in the cage. So the mouse is anesthetized, and you can see that there are mosquitoes here, so they will bite the mouse and they will get the proper natural infection. So this is the infection with the sporozoites. But also, I mean, most of the lab, because this is very tough to maintain the mosquitoes and it's very tedious job. But also now these days we can buy sporozoites from the respective institutes. But there are also blood-borne infections of the rodent parasite strain, which I will show in details. That is also possible. So the need of the cerebral malaria model, as I was mentioning, that we can get terminal event data, right? The patient who died of cerebral malaria, we can get the histopathological data from the brain tissues and the other organs. So there are real-time data we need. So that's why we need the mouse model also. I mean, but in future, those will be obsolete also. So this is a small figure showing the utility of the mouse model into the basic research. So here we have two mouse models. The B6 is the wild type, which dies with the cerebral malaria around 7 to 8 days post-infection. So post-infection means after we infect the mice at 0 day with the rodent strain, which is called P. burgerianca. This mouse model dies, all the mice in this group dies around 7 to 8 days post-infection with the cerebral malaria, coma, seizures, and septic shock. But this is a mouse, the dashed line, it survives the infection. So I'm not going into the detail of this strain, but this is a resistant to cerebral malaria mouse. And after we infect this at 0 day, this mouse surpass this phase and they don't die with the cerebral malaria, and they die with the hyperparasitemia at 30 days post-infection. So the utility of this two mouse is to understand what is the difference between what is going on here in the resistant mouse compared to the wild type. We can see with the parasitemia, which we read from the blood with the Gimsa stain from the tail smears, and we can see that as this mice, the black line, they die at 8 day post-infection. There is no such huge difference with the parasitemia kinetics, but here we can see in the one which survive with hyperparasitemia, the parasitemia is increasing and they die with hyperparasitemia. So the mouse model help us to reveal the what is going on with the infection, and then we can correlate this message or translate this message to the human, and the therapeutic part can be approached there. So this is a graphical representation at 7 day post-infection with the parasitemia. So as this is the peak phase, the 7 to 8 days post-infection, where the mouse shows in the wild type the peak of the coma. So there we can see subtle difference in the parasitemia. This is just a representation. And then we can see that the spleen at 7 days post-infection in the wild type mice is much smaller. So this dashed line is the control baseline. So summary of this two, and the infected mice spleen is this, and this is one representation of the infected spleen from the resistant and the wild type mice. So we can see that there is an increased splenomegaly is happening. So the spleen is working properly. It's capturing the infected parasites and the debris. And this is very nice presentation actually. If this is a control brain from both the mouse, and this is the baseline, the dash one. So what we do here, we inject the mouse with the Evans blue dye, retroorbital, during the infection, during the peak infection. And after 45 minutes, 1 hour, we sacrifice the mice and we see the dye leakage in the brain. So if there is a blood-brain barrier disruption in this mouse, which is there, so the brain will turn blue due to the dye leakage. So if there is no blood-brain barrier disruption, there will be no leakage. So you can see clearly here with the picture that there is no, so though we injected dye in this mouse, it didn't reach the brain. So the blood-brain barrier was intact. So this is the usefulness of a mouse model. This is just a representation. And this is a measurement of the dye, Evans blue dye in the brain. We can see there is a huge concentration in the wild type mice compared to the resistant mice. And this is a schema showing what is happening. So this is accumulation of lot of data and try to show in a summarized way. In spleen, what is happening? There is increased IFN gamma, decreased IFN gamma, CD4 T cells, CD8 T cells, chemokines. And in malaria, what happens that there is infiltration of the T cells in the brain with the help of chemokines. So these are CD8, CD4 T cells, and the cytokine also infiltrates into the brain. And as you remember, I was mentioning the blood-brain barrier disruption. So this, and because of the parasite sequestration, and it leads to the infiltration of the CD4 T cells, CD8, chemokines, and the patrolling monocytes. And in brain, what happens inside the brain? We can see that there is a activation of monocytes, microglial cells, astrocytes. So these are the microglia and astrocytes, the brain resident cells. And we can see that they have respective markers, activation markers, which showcases the fact that it's leading to edema, swelling in the brain, inflammation, causing the experimental cerebral malaria, and finally leading to death. But what is happening in the resistant mice? In resistant mice, there is a retention of the stuff in the spleen, increased MCP1, IFN gamma, IP10, chemokines in the spleen. And we can see that there is inhibition of the migration of T cells in the brain, and also less parasite sequestration in the blood, and which is leading to the reduced blood-brain barrier disruption. And we can see with this red crosses here that there is a reduced activation of microglial cells, astrocytes, less edema, swelling, and we saw that with the with the brain picture that there was less coloration. So all the stuffs are happening there inside the brain. It's tough to show all the data in a graph wise, and there is reduced inflammation also. But there is one thing which you can see, that the CD300F, which is not present here. So this was one of my study where I showed that the presence of the CD300F on the microglia, which is a transmembrane receptor, is important requirement for this mice, which may lead to the protection in this mice. So this was possible because we used the mouse model, which was resistant to some stuff, and further studies are needed on this to understand the downstream mechanism. So after this, I will explain something on the toxoplasma. I mean, why? Because this toxoplasma infection is mainly carried by cats. And as we know that the cats are fond of the stuffs, and it circulates among them, and it leads to the release of the oocyst, fecal oocyst or tissue cyst, which are then again consumed by the respective animals and then to humans. And in this way, the cycle continues. And in India, I don't know how much prevalence of toxoplasma is there, but in Europe and USA, obviously there are prevalence. But there are two phases of toxoplasmosis. One is acute, one is chronic. There are other strains, and also there are mouse models to study those stuff. So this is interesting that in this infection also there is a role of central nervous system in this. And as I was mentioning, that this is an intact brain parenchyma, where there is a monocytes, astrocytes in brain tightly held. But in toxoplasma, which chronic infection, which I will describe, not the acute. In acute, it's very severe. Here also, they invade the central nervous system, increases the permeability of the blood-brain barrier. There is a moderate inflammation though, but CD8 T cells, the activation of glial cells, most of the mechanisms are common than the malaria, which I was mentioning before. Then what's the difference? I mean, why I am describing this two together? So there's a tricky part here. So we in France, when I was working there, we developed a model there where the pathophysiological modification possibly induced by chronic toxoplasma infection prevents experimental cerebral malaria induced by the rodent strain. So here we use the mouse model, where we injected the mouse model with the toxoplasma strain, rodent strain, that doesn't hampers the human system, and we check the survivability. So we were very happy with this data, that when a mouse was, this yellow line, if you can see, that when the mouse was infected with toxoplasma, and then after a certain period of time, which was experimentally proved, we infect the mouse, which is already infected with toxoplasma, with the malaria mouse, they don't die with the cerebral malaria, they pass this phase. So what does this shows? That there is a increased survivability in a mouse model if the mouse is already infected with toxoplasma chronic infection. And these are the other groups where we have sulfadiazine, which is a control group, and the treated group, they all die. If you see with the PEVIA B6, means the Plasmodium burghianica, the green, red, and the purple, these are all, this is the wild type group, and these two are control group, and this is the co-infected. So this gave us some idea that now we should proceed further what is happening in the brain. But there was no difference in the parasitemia, so which is a good thing. So parasitemia has no role in this increased survivability in the co-infected mice. So there must be something going on there. So we did the experiment again. I mean, I'm showing just the basic stuffs just for the clarity. If you have any questions, please ask me in detail. So this is a control mice where we took the brain at the respective time point after injecting the Evans blue. This is the wild type B6 mice, where we have seen before that it turns blue because there is a blood-brain barrier disruption. This is a chronic toxoplasma infected mouse where there is no blood-brain barrier disruption, but there are infiltration of T cells in this mouse model which are known before. So, and this is a co-infected mice where there is infection of toxo, and then the mouse was infected with the lethal malaria mouse strain. But you can see that there is a very decreased blood-brain barrier disruption, which is indicated by the less blue coloration. So that means this is one of the reasons that presence of Toxoplasma is helping in reducing the blood-brain barrier disruption during malaria infection. So we then extended this study, which I'm not explaining here, but we found this nice mechanism that this is all happening in the mouse, which is co-infected with Toxoplasma and then infected with malaria. So our hypothesis was there that something like this might also be happening in nature, because in nature we speak about co-infections, a lot of co-infection morbidities, and there are a lot of areas involved where, like Amazonian river basin and all, we have infections like Toxoplasma, Leishmania, malaria. In India also, like in Bihar, we have malaria and Leishmania both. So here we can see that the cyst is present in the MSC microglia, and there are processes like autophagy and all. So the presence of cyst suppresses the parasite sequestration via innate lymphoid cell 2, which leads to the decreased CD4, CD8 T cell infiltration. So if you remember this mechanism, this also we observed in the resistant mouse infected with malaria. So there are always some common mechanisms. So we also hypothesize that maybe the T cell exhaustion is happening, antigen presentation issue is there, and also monocytes might be playing some role in this protection. So further moving on to the last topic, what is the need of the basic and translational human immunology. So as we know that, I mean, the translational human immunology is a very important aspect now, because the tools for basic stuff is very important. The blood-based immunoassays, report analysis, modeling immune tissues, like tissue-based immunity, organoid like cell culture, and in vivo studies like natural immune variation and in silico models and bioinformatics. So these things were on the verge of coming out, and we saw that in last 1.5 years, science is moving like anything, because the accumulation of the information from the sample and using the in silico and bioinformatics application was very useful in last 2 years. And I think maybe I'm wrong, that the already available information in the scientific community and the tools help us to get the whatever efficacy the vaccines has. I mean, it helped us to understand those things. So these days, the wealth of the human data for translational immunology is very important. We have several cohorts. Cohort means the human patients, and also we use control groups. So we get samples from those cohorts. We get a lot of other data, which help us to store specimens and used batch-wise respectively. Like we get blood samples, primary cells, we get fecal or urine samples. So what information this samples or the cohort gives us? So they give us functional information, specificity, the tetramer-based knowledge, VDJ data, and also they give the informations on the phenotypes of the cells present by using flow cytometric applications or RNA-seq information. They also give us information on the intracellular cytokines, like application of mass spec level. They also give us the functional aspects. And then more interestingly these days, I mean, from this samples, we also get the information of the gut microflora and the bacterial occurrence in the respective patients, and how accumulation of all this information through the approach of the ex vivo and computational analysis in vitro models will give us such a huge information, which will be the future of the research. Because before, what we were doing, we were mainly using the basic discovery and mouse model stuff due to those were the need of the hour that time, and also we were validating in the non-human primates and then testing in humans. Then presently what we are working on is we are gathering sample and data from humans. We are working on animal models and the in vitro models, collecting data and information, and then correlating and predicting with the bioinformatics. But I think more we will work on this stuff in future. There will be less and less use of the animal models because of the ethical issues. It's tough to get clearance for the animal work because of their because of the guidelines of respective institutes. So in future, we will work more mainly on the mass cohort collection samples, and those will be very crisp and well planned, because people will have more experience on those things. But for sure there will be use of the mouse models for the better clarity, but the number will significantly decrease. So in summary, reemergence may happen, because of a breakdown in public health measures for disease that were once under control. And I feel that co-infections may promote intricate and complicate mutualistic interactions with the host system, and that might lead to even a pathogenic outcome, which I didn't explain here, but also protective outcome that need to be studied in greater detail in respect to the geographical locations of the infections, and also respective vectors which are co-infecting. But there is no quick and simple experiment that can firmly determine whether immunity will be effective or lasting or protective or pathogenic. It is just soon to know in case of infectious diseases. Though for several decades we are working on this stuff, but still a lot to know from this stuff. So these are my previous labs. This was when I was working in Institut Pasteur, Lille, France, after finishing my PhD in India. And then I worked short period in Nice, France. This was from 2000. During the COVID phase, I was working here mainly on malaria. I didn't explain the recent works. It would have been very lengthy. And then currently I'm working here at Harvard and MGH. I'm working here mainly on the antibody engineering stuff, and working on affinity and avidity stuff. But the topic is food allergy, which I think I got opportunity to work on something where in India it's still a new field. Hopefully things will go better. So I would like to say thank you everyone for your patience and time.
Thank you, Tarun, for such a wonderful and informative session. So we understood, first of all, how the microorganism, how the microorganism has a constant struggle to survive and emerge and re-emerge as an infectious disease. Comprehensive details of such major emerging and re-emerging infectious disease were presented since the year 2000 today. Tarun also has discussed on the pan-Ebola vaccines and their mechanism of action. And finally, he had discussed on the glimpses of host pathogen responses that are orchestrated in model systems such as malaria and toxoplasmosis. And finally, some importance on the translational human immunological research as an alternative therapeutic opportunity for future human use. So it was such a wonderful session and informative indeed. So before going to the question answer session, I would now like to request all the participants who are watching us online to fill up the feedback form for today's session. It will be available in your chat box. So now I would like to go to the question answer session. So first question is
Tarun.
Yes, sir.
May I ask a few questions?
Sure, sir.
Okay, so first, I would like to thank you for your very lucid yet very comprehensive and scientific presentation. Okay, Tarun, very good job.
Thank you, sir.
I have two quick questions. First of all, you talked about the pan-Ebola vaccine where glycoproteins, the Ebola glycoprotein is being used as the material for vaccine. And you showed that its storage temperature is 5 to 8 degrees.
Yes.
And in a poor tropical country like Africa, where people are mostly affected by this virus. And now that we know during this COVID period that different vaccine platforms are being used, like mainly the DNA vaccines or the adenovirus-based vaccines, where the storage temperature can be a bit higher and it's a bit easier to store. My question is, do you know about scientists who are trying other platforms of Ebola vaccine where it's not the glycoprotein, but like DNA and other vaccines are being used?
There are other groups also working on other stuff where the storage temperature was not like this. So this formulation was based here after there were several experts who already had the prior experience on this vaccine stuff. So the good thing about this was that the storage, but it's a good observation. I mean, even during the presentation, I missed this part that we had this advantage with the temperature. But there are other groups who are working on the other formulations of the Ebola vaccines, and frankly speaking, the GSK already got license for the vaccine, Ebola vaccine, GSK, and I think there is another company. So they got actually the license for emergency use in 2019 because there was a huge outbreak in 2018-19, and then something again happened in 19 mid. So they got the approval for the vaccine. So as you know that if suddenly some company gets the vaccine and other, then the funding for the other who are already working on those get little bit crunched. I hope I was able to answer your question.
Yeah, very much. And Tarun, let me tell you that, these African countries, Congo, Zaire, Kenya, Tanzania, these are very popular wildlife destinations. And lots of people from Calcutta and India, they go for wildlife photography. And I am also planning to do. But I tell you that before going to Africa, yellow fever vaccine and Ebola vaccine is a must. Anyway, my second question is very tempting. Okay. You talked about the cerebral malaria in details. And you talked about the mice model, and you showed that this BART2WLM mice is a resistant mice, right? So it is true for many epidemic and pandemic diseases that the entire population never gets affected. There are always people who stay like,
healthy.
what to say? I mean,
carriers.
Asymptomatic or carrier, whatever. And these days people talk about genetic predisposition, and for that most of the time SNPs, the single nucleotide polymorphisms, are responsible.
I think there are a lot of labs in Kolkata also. They are working on SNP.
And because you have the detailed immunochemistry done on your subject, like the involvement of CD300F and others. I mean, it is very tempting. I know you cannot leave your line of work. I mean, think of collaboration. Okay, if somebody can study, people do GWS, genome-wide association study, to see what are the SNPs responsible for a particular trait.
Yes, I mean, I think in last few years of my PhD, I tried to get the human samples. It's tough to get things.
Right.
So it was my plan to work on human samples also, at least on the PBMCs, to get the immunophenotyping on patients, because in Kolkata we had that time a lot of patients and sudden malaria and outbreaks. But I was never lucky to get those samples to work. But it would be great opportunity for me in future if I can work on this.
Okay, I think thank you, Tarun. Thank you. I mean, if others have questions, please come out.
I have a question.
Yes, sir.
Tarun, I think there is a great similarity of our acquaintance. I don't know. You have done your graduation from this college, and I'm the principal here. And I actually did my entire graduation, post-graduation, and PhD. I have done it in KESHWANI University.
Okay.
And
sir.
later on, I worked with.
If I can add one thing, sir, I went to Kalyani because I wanted to study in university, and marks were very low, so I didn't got in premier institutes in Calcutta.
No. That is not an issue, but you have ultimately come off that initial problem.
You have crossed all of them who got better marks than you.
I mean, but those things matters.
Another thing is, I have worked under ICMR, that is Malaria Research Center. I was the senior scientist in Chennai Field Station in Chennai. So I had a lot of experience in organizing vector control and other activities in malaria.
Okay.
And actually, malaria, that is a national NIMR, that is.
I know a lot of scientists in NMI also.
That was my actual. That was my mother institute initially. So I worked under them, and still I have contacts with them. But the thing is, very interestingly, you said about the vaccine possibilities with malaria and parasite. But my point is that you see, normally what happens with the viruses and other microorganisms, there is a lot of variation, and not all the vaccines which are produced are equally effective in all parts of the world.
Yes.
And when we talk about the malaria parasites, there are a lot of variations also.
Yeah.
And I feel there are a lot of stereotypes, that there is a lot of differences are there present, and we too have found a lot of other sibling species who are involved in the different spread of these diseases and all. I just wanted to know from you that whether these entire issue will this vaccination prospects will be zone specific or area specific, or it can be wide spectrum the vaccination can be done.
So if you ask me, I think the vaccine thing can be solved if people work in collaboration, because people are working on different stages of the parasite, and it should be multipotent, because a single site vaccine will not work in malaria. I mean, and in malaria we get the information about a patient when the parasite is already in the blood stage, so it already entered into the liver, and then we are seeing in the blood. So by that time, when we are observing the parasite in the blood, when the patient is responding to the medications, like they have fever or something, or sweats, we don't know whether the parasite is already in brain by that time or not, or it's too late. So I think multiple effective site for vaccine we need, but for that we need a huge collaboration worldwide where people have respective expertise.
But you are working with Plasmodium falciparum, isn't it?
Yes.
But you have not started working with Vivax or other.
No, I didn't study Vivax stuff, but I know in India there are some institutes like BITS Pilani. There are people who are working on Vivax, and they also showed that in Vivax there are also severity. In brain also,
yeah,
the Vivax
severity is there, but Vivax normally that's not very deadly as it is said to be.
Yes, but they found few cases of Vivax which are deadly. So I mean, Parasite is also evolving. So I don't know detail about that, but I think the vaccine is possible, but we need sharing of information. I mean, then people, for those who are formulating vaccines, it will be easy for them to understand what is going on, because for each it's tough to understand all the phases of the life cycle. This is huge task. But your question is very relevant, sir.
But I was really related to understand that you are working on the malaria vaccine, and that is a real need of the hour when we think about the entire global scenario. Thank you very much for your talk.
Thank you, sir.
And really it has excited me.
Yeah.
Thank you.
Thank you, sir.
Okay, Tarun, we have one last question from a student of our third year, I think, Rishabh Kundu.
Okay.
His question is, what is the role of melatonin in malaria pathology, and what is the role of cerebral malaria pathology? What is its role in cerebral malaria pathology?
What is the role of melatonin in cerebral malaria?
Yeah.
I mean, I don't have the right answer now. I will go to the second question first. So what is the physiopathology of, what is the role of cerebral malaria?
Yes,
is that the question?
Yes. What is its role in cerebral malaria pathology? So basically,
I mean, there are multiple factors which have impact on cerebral pathology. I mean, it's tough to say precise role of melatonin in cerebral malaria pathology. I mean, it's multifactorial actually.
Right.
So I mean, it's a very thoughtful question though. I mean, but you know, I mean, I have no answer. What would be the? I mean, if I have that answer, I would have published a paper by now on this topic. But I mean, but a crisp role on that. But in general, the cerebral malaria is the outcome of multiple factors going on the host. That is the end point when the mouse has the cerebral malaria symptoms. Before that, a lot of damage has already been done. I mean, including multiple organ failure, which I missed to mention.
Right.
Yeah. But Thank you for the question. I mean, Rishabh.
Yeah, we will definitely look into it. Okay, we have almost come to the end of our today's session. Now, I would like to request Professor Nilanshu Das, the HOD of Molecular Biology Department, to deliver the vote of thanks for today.
Okay, I think I'm audible, right?
Yes, sir. I mean,
It's my pleasure and privilege to cast vote of thanks today on behalf of the Department of Microbiology and IQAC of Surendranath College. Teachers are lived by their students, and it brings extreme satisfaction to see our students in frontline research institutes of the world, like Harvard Medical School. I won't express today my usual gratitude that I usually express for the speakers, but this time I'd rather thank TARUN for giving us your valuable time today. It's a Sunday weekend, but still you spend your time with us. We definitely count you as one of our golden alumni. I'd like to thank all the participants from all parts of the globe who were virtually present with us today. I express my gratitude to our principal, Dr. Indranil Kaur, and the governing body of Surendranath College for being a constant source of encouragement. Thanks are also due to all the members of IQAC and our technical team for their crisp management of this webinar. And I conclude by expressing my love, respect, and thanks to all my colleagues of Surendranath College, both teaching and non-teaching, for being with us throughout this webinar. Thank you all and stay safe. Thank you.
Thank you, sir. Thank you for the nice words.
Thank you, Tarun.
Thank you.
So this brings to the end of today's session. We will be bringing you a series of new sessions from the desk of scientists and physicians who are directly involved in the research in the diverse field of biomedical sciences. So stay tuned and do join us on our upcoming webinar and podcast. With this, me, your host, Dr. Devjit Dasgupta, signing out. Thank you.